We move on to Ansgar Petersen, who was already mentioned, as well as his topic of cellular biomechanics. So your talk perfectly set the stage for his. Now, Ansgar Petersen has the professorship for Architectured Biomedical Materials for Tissue Regeneration in our section of Advanced and Personalized Therapies. Important to note is that Ansgar studied physics and finished his PhD in biomedical engineering at the famous Technical University in Aachen. And he also had a company experience as a development engineer, but then moved back to academia and joined the Julius Wolf Institute at the Charité, now belonging to the BIH. Since 2012, he was head of the research group on cellular biomechanics there, and he focuses on defects in musculoskeletal tissues in consequence of trauma or other events. As a biomaterials interested person, he's also interested in the interaction of materials with cellular function, and this is one of his original fields of expertise. He designs also on purpose microenvironments to provide specific signals, either mechanical or also biological signals to improve healing. So Ansgar, we are highly interested in your talk, and now the mic is yours.
Thank you very much for the kind introduction. It's my pleasure to take you now towards the field of bone regeneration. So in general, in my group, we are interested in understanding how mechanical and geometrical signals influence cells in their attempt to regenerate tissue. This manifests often in biomaterial approaches, and this is in the musculoskeletal field, but I'm very interested also in applying these principles in other fields. So I'm looking forward to talking to you either after the presentation or in future about potential collaborations. So I'm quite sure that most of you are aware of the fascinating regenerative capacity of bone. If you ever had a fracture, you know that after a couple of weeks, the bone heals and restores its full function. But in a situation where a larger piece of bone is missing, as you can see it here after tumor resection, this is a more severe problem. And how this was approached here was by doing a segmental transport of bone, but in the end, the bones did not fuse. And this would require an additional surgery where you again apply autologous bone material from the patient, or you apply the biomaterials and growth factors to try to address that problem. So one of the questions we are asking in the group is, can biomaterial architecture have an active role in bone regeneration? And why we believe this is an important question is, if we would be successful with such an approach, this would have a lot of advantages. We could produce a material that can be available off the shelf, it can be cheap, and it can also be safe. And with that, it could support the surgeon's job in trying to find solutions for these severe bone defect situations. If you have a look at what has been done in the field in terms of biomaterials, it's surprising to see that there is a large number of approaches which follow a biomimicry idea. And the tissue that people try to imitate is the spongy bone that you can see here, so the trabecular bone, which seems to be fascinating as a motivation for biomaterials. And this has been done quite well. For example, you can see how you can use a forming process to produce similar structures from polymers today also from metals. You can create artificial designs which look like trabecular bone and also optimize the mechanical properties, something we've done here in-house, but also which is done in companies. We can also, and that's a large portion of research being done, also try to imitate the chemical composition. Bone is mostly composed of calcium and phosphor. So a number of biomaterials are following this idea to create the according chemical environments. But also you can combine different approaches here, for example, the chemistry in terms of calcium phosphate particles combined with a flexible polymer to create these sheets which you can bend. This is a commercial product available, as most of the products I'm showing here. The problem is that if you implant such materials alone, you realize that they are not very effective. This is an animal model we use to study bone healing. You can see on the left an untreated defect. A little bit of bone is forming here, but the main part of the defect remains unbridged, so there's no tissue forming, no mineralized tissue forming. And it looks very similar if you implant commercial biomaterial. And this situation is indicative for that the role of biomaterials today is actually to serve other bioactive components. So you could say these components like growth factors and autologous cells, these are the original active components, and the biomaterial serves as the carrier thereof. So coming back to the question, what architecture can do, the problem we are facing is that there is actually no blueprint we could follow, no tissue we could imitate to achieve healing of such large wounds or defects. So how we try to address this question is by reducing the complexity of the geometric situation. If you look at the trabecular bone, each point on the surface can be represented by two principal curvatures. And from these principal curvatures, you can create, again, very simple geometries like cylinders and spheres, which can be either convex, so hill and rim-like, or concave, so pit and valley-like. And following these ideas, we, in the last years, developed a cell culture chip, which is based on a molding process. So starting with a micro-machined brass mold, we also use a sacrificial sugar mold in between. And in the end, we are able to produce cell culture substrates, which are have a very high surface smoothness and very defined geometries. As you can see, multiple geometries that we can now use to study how cells respond to these surfaces. So you can see here this chip, and while I zoom in, you can see the different geometries we have realized on the surface. And I want to show you one specific section where you see confocal microscopy of cells on these surfaces, cells that were stained. And if you carefully follow, you can see already differences between how cells behave here on the rims and in the valleys. For example, if you follow this slow migrating cell, when it enters the valley, it suddenly speeds up and is faster. Other cells try to bridge these small gaps we have here, these valleys. And that is observations that we can also quantify and which we have done in the past. To summarize the work we have done on these geometric chips, which we call geochips, we found interesting cell responses. For example, on spherical convex surfaces, bone progenitor cells tend to differentiate towards more mature bone cells. On convex cylindrical substrates, we have a very robust guidance of cell movement and migration, which is actually competing with nanotopographical cues. In concave environments in general, we have a higher cell mobility, but especially also in the concave cylinders, we see interesting mixture of cell alignment, this cell spanning I've shown you before, and the increased mobility. And I show you a couple more pictures in a second. Interestingly, all these effects take place once the diameters of these structures are reduced down to a couple of hundreds of microns. So 300 micrometer is a typical size that is very instructive. This is in the mesoscale, as we call it, and the according geometries that we identified we regard as architectural building blocks that we can in principle put together to build more complex pro-regenerative biomaterials, and that's something I'm very interested in following more in the future based on the work we have done so far. So coming back to these valley-like concave cylindrical surfaces, if you now have a look here at these confocal images and the reconstructed cells, if you go from bottom to top, you will see an increase in curvature. And what you observe is that more and more cells tend to lift off and span over this valley. And we've understood that this is a property of certain cell types, but not all cell types. We know this for fibroblasts, mesenchymal stromal cells from the bone marrow, but also osteoblasts in contrast to certain cell lines or many cell lines, and for example, endothelial cells. This is a consequence of the specific cell physiology, especially the distribution of the focal adhesions here at the ends of this elongated cell. So they seem to be made to close small wounds, and that's what we believe is not only, as described, relevant for embryonic development, but also for healing of micro wounds which might occur during daily activity. So if you look closer now into full cylindrical pores, you see that the same principle is applying. You see it here fibroblasts, and see how they first form a layer of tissue until they reach a certain curvature or according diameter where they are able to cross over to suture, as we call it, this wound. And then this wound heals very fast. And because it's a loose network, allows also structural remodeling, and cells align in the direction of these channels. You can see it here also in the cross section, the fast closure of the wound, and then here the according alignment. So in principle, we can use channel-like pores to allow cells to migrate fast, to form tissue very fast, and to align. And that's why we believe, or why we thought that this might be a good geometry that could help us tackling the problem of these large bone defects. What you can see here is insights from an animal model, where we can observe how after we set the defect, tissue structures form. So you can see white collagen fibers, and you might appreciate that there's something like a scar forming at the end of the bone. This is second round imaging showing collagen fibers, which is then also stabilized by a bony shell forming and closing, enclosing the bone marrow. And that's actually what we not want to have. We need these cells to form new bone in the defect. So creating a structure that has a cylindrical general shape in the appropriate dimension, and that guides the process of bone formation from one side to the other, is something we wanted to apply here and see if that improves bone healing. So we are looking for a technology to do that, and I'm since many years in contact with the company Matricell, who have this unique technology to produce aligned porous biomaterials by a directional freezing process. So you can see here ice crystals growing from a colder to a warmer side, and if you do this right, you can end up with these finger-like ice crystals, and after freeze drying, this can leave behind biomaterials that have a channel-like pore architecture, actually exactly in the dimensions that we are looking for, around 100 micrometer. It's a spongy material which you can tune, so you can change the stiffness, and you can also change the pore diameter. And these have been studies that have been ongoing over the last years to optimize this material to be effective in recruiting cells and also to differentiating cells, or allowing cells to differentiate towards bone. But if we talk about a biomaterial that's supposed to support bone defect healing, this sponge-like structure is too soft, and we came up with a design concept which is such a hybrid material containing the direct cell environment in terms of the soft biomaterial and the stabilizing frame, which is also specifically architectured. For this, we collaborate with the Fraunhofer ILT in Aachen to produce this from medical grade polycaprolactone, so a resorbable polymer that in the end the whole product can be, or the whole biomaterial is implanted, then fulfills its function and degrades over time, and nothing is left over. Of course, we have to verify the mechanical function of the material. You can see here how we use our mechanobioreactors to apply in vivo like loading conditions and check for material fatigue. This led to a couple of design optimizations, but in the end these structures are now stable over millions of cycles, and we believe are very suitable for implantation. The material that we developed is very easy to handle. You can see how it's easy to wet. You can use it like this now for cell culture experiments, but also for implantation, which would be exactly the same case. We wet it in a buffer solution, and then we implant this material cell-free with the idea to recruit cells from the bone marrow. And that the cells are indeed, or the material is indeed able to recruit cells, you can see very nicely here in this time-lapse microscopy video from the in vitro culture, mesenchymal stromal cells entering the material. And what I would like to point to is that the reason why these cells are moving so fast is that they again are doing this spanning in the architecturally optimized environment, which allows them to migrate very fast. And here, this is another example where we placed a cell spheroid on top of the material, and after 7 days, you can see how many of these mesenchymal stromal cells we have recruited into the depth of the material. This is important because we have only a certain time window in which we can recruit cells from the adjacent tissue, here especially the bone marrow, to fill the material and then to allow bone formation. So this is the jump towards the in vivo situation. If we implant this material, you can see that we are indeed successful in obviously recruiting cells that are now present all over the material, which you can see in this region. And we are also able, or the architecture supports the alignment of the cells, which leads now to a situation where we don't have a scar formation as we had in the empty defect, but we have collagen fibers connecting actually the two sides of the defect. And you can see already certain maturation processes going on here I'll show you this more in detail in the next slide. So this is the situation I've shown you before for an empty defect and also for commercial material. And the picture I show you now is the result of many years of work. It looks very simple, but indeed, until we got there, this was quite a long way. But it was very successful. As you can see here, we are able, this is a picture after 3 weeks, to indeed, in this hybrid scaffold, recruit the cells we need from the bone marrow. They are serving here as a reservoir, and we see how then there is a spatial temporal maturation cascade, which forms first cartilage and then bone. I must admit that we did not intend to induce this process of bone formation via cartilage intermediate phase. That was a surprise and a very relevant finding for us. This is especially interesting as this process mimics very much the process of bone formation in the growth plate. So during childhood and adolescence, this is how a lot of bone in our body is formed, a very controlled process of chondrocytes going into hypertrophy and then forming this here black stained mineralized tissue. And we see very good similarity to this process to what we have induced using the biomaterial, especially by aligning the process and preventing the interference with surrounding tissues. So in principle, we have created a niche where bone can heal, and that this is a robust process of producing bone. You can see here now after 9 weeks, the process continues. We are now in the phase where the two sides start to bridge. You can see collagen fibers in yellow already merging over here, and this becomes even more clear if you look at the micro-CT data, indicating again an earlier time point and a later time point. In the early time point, you can actually see how in each pore of this sponge-like material, a little bone piece is growing. This is an indication for us that this is an active process which continues, and we are now here at 9 weeks in the process of bridging, as I told you. This is another representation, and you can see how the gap distance is decreasing over time. Now we are here with these animals already in the phase of bridging, and this is to us a very motivating result, and it's really a breakthrough because we use only a pure material approach. Of course, we want to bring this further, and the next is the proof of concept in sheep, which is planned for the beginning of next year. For this, we had to scale up the material. You can see here again the 3D printed support. And now the final product, which was already waiting for surgery. And we also want to employ or investigate if this material is suitable to heal osteochondral defects in the knee. That's a big problem still today, the restoration of the bony, but especially also the cartilage structure. Why we believe this might be interesting is because we induce through the biomaterial form bone through a cartilage intermediate phase. So maybe we can achieve a synchronous restoration of both structures through one material. And I want to thank the people which have been involved, the team. Some, it's a bit older picture, some have left already, some new members joined, but it's a very nice atmosphere and I enjoy working a lot with you guys. The clinical partners, Georg Duda, of course, our industrial partners, especially Ingo Heschel and Hans Lehmhuis, we have been working together with for many, many years. Also from the Fraunhofer ILT, the colleagues and the international collaboration partners. I want to thank you for your attention, and I'm looking forward to questions if you have any.
Thanks a lot, Ansgar. I haven't seen any questions coming up, and I'm afraid we've already run out of time, but you gave another very convincing presentation. So I would conclude that high fidelity is reaching regenerative medicine. That's very good to see. You mentioned the importance of fidelity, and the data that you've shown are really promising to solve an important clinical problem. That is true for everybody who presented today. So I'm proud to have everybody of you on board. And there were even 2 more recruitments in this year who couldn't present today. The one is Sylvia Thun, full professor for interoperability and digital medicine. She already gave a BIH lecture in spring. And another person who we are missing today for a very simple reason, he's on holidays, is Tobias Winkler, who makes a very important contribution, as already indicated also in the talk of Ansgar Petersen. in the field of musculoskeletal therapies. So he will certainly get an opportunity to present his work then in the next year. So again, many thanks to all the speakers, to the audience for staying with us. I think everybody had a very interesting afternoon and late morning with these presentations, and I look forward to seeing all this growing together in our institute. Okay, and then with that said, goodbye, and also many thanks to the organizing team once more.